Test, test. Is my voice clear?
Yes, sir.
Okay, so good afternoon, everyone. So let's start our third session on bioinformatics computation. This is computational course for you to be able to work with genomic dataset. And I think there's another class you have for much more on the bioinformatics side. So this is a complementary for you to make sure that you are aware and capable of using platform that are available in the market. Okay. So last week, I believe you already learned about Terra. Make sure that we spend enough time here. Maybe today, if there is additional demo from Calbe team, you can use. We can continue Terra. So Terra created by many big companies like Microsoft and Broad Institute. So you can get your free credit from GCP, Google Cloud Platform, and then you can use it at least for $300. If you need, if you want more, you can subscribe yourself for your research. And in this Terra, you can get some workspace or workflow that is available from Broad Institute, usually with the Illumina and GATK library. So this is a good tools for you to get started with. Okay. So let us cover the material first. This is about how you get data. And after that, we'll continue with demo. Please practice because the result of your education is on yourself, on amount of time you spend on the tools, framework, and books. Okay, so bioinformatics and genomics start from a central dogma that we adopt until now, that from molecular biology, if you go down into very microscopic dimension, you will see inside the cell, chromosome, you will see DNA. And DNA is actually producing RNA, yeah? And RNA producing protein. And it will impact our phenotypes through our metabolites. So in terms of omics science, not in molecular biology, they call it DNA, they call it genomic. RNA is about transcription of DNA, so they call it transcriptomics, yeah? And protein is about proteomics, and metabolomics. This study have been very active for a long time. It's not new, yeah? So it's already a long time. But the impact in the industry, we're still looking forward that we can learn about our DNA, RNA, proteins, and how it impacts our phenotype. Like, for example, can we drink more coffee? Is it good for our body or not? Or can we drink more milk? It's all impacted by metabolites, proteins, RNA, and DNA. Okay, so that kind of testing, like nutrigenomics, is available in the market. But there's many other testing like skin genomic, cancer genomic, other genomics, or maybe if Indonesia, we call it agriculture country. What about agrigenomic? What about if we can use DNA, RNA study to learn what kind of natural phenomena that creating, for example, palm oil, palm tree that can produce shorter palm tree and more resistant to climate change. There's so many applications of this central dogma that need more bioinformatician to work with. So if you learn in University of Indonesia this time, I hope it can inspire you to explore more in this area, because the future of technology, the big future is on digital biology. Okay. What we see in digital now is just apps, AI, data, but the real complex biologic team, digital system is our own body. So looking into our own body through digital biology is one of the future area that I think worth to invest. So in terms of tool set, so if you read functional genomic books in the books folder in the Google Drive, so initially all of the researcher working with the web-based graphical user interface. So many portals like NCBI, EBI, DDGB from Japan, EBI from European, NCBI from US, and many tools like genomic browser is available. Many graphical software like Mega, RStudio, Biomart, and many other tools like Galaxy is widely available. But on other side, there is also some researchers that they don't like to use web-based interface, but they much more like to use command line interface, like in Linux. They just want to code. They don't want to get distracted by the user interface. So in this case, many other frameworks from Python ecosystem like BioPerl, Python, ByPython, etc., is also widely available. So you can learn all of this both from web browser or from command line, but you have to be very opportunistic in your case, because if this is available in Galaxy, for example, and then use Galaxy. If this is available in Terra Bio, then use Terra Bio web user interface. But if only available on, let's say, SnakeMake or Nextflow, then you can use SnakeMake and Nextflow. So here in the table, I took it from the books. So there's many web-based tools listed there, like to access information, you can use Biomart. For pairwise alignment, there's a BLAST library for command line. Okay, database search, all of these tasks, you can see all of the library here. Unfortunately, it's very fragmented. So you have to choose what is the right one for you along your journey in learning bioinformatics and genomics, because there's very fragmented tools over there. In this course, we will much more focus on Terra Bio. I hope today you explore more about Terra Bio, but after that, we will go into Galaxy project. So Galaxy project is funded, I think, from Australian or community, or and you can use the pre-install Galaxy. So this is drag and drop tools. So you can get the data from here, but you need to learn it step by step, because so many of you learn at the same time. So later on we can learn Galaxy, but for today, just focus on Terra Bio first. Maybe next week we can learn Galaxy or other library. For command line, you can start to take a look on Nextflow. This community is very big in Europe. So in Nextflow is a command line based on scripting language like Java or Groovy, yeah. So you can also reuse many sample of workflow here in NF Core. So it has some like repository of workflow built on top of Nextflow. Other library that is also popular is SnakeMake. So SnakeMake, Nextflow, Galaxy, and Terra will be our focus to explore and hack during this course. Okay, SnakeMake, this is SnakeMake. So if you read papers and the papers provide code in SnakeMake, SnakesFlow, or Galaxy, that's good. You can reproduce if they give you the source code. And I think like in AI, most of the researchers are very open. When they publish paper, they also publish the pipeline. Okay. So next, what are the tools you need? For very basic tools, I think this one is good, Orange, that I think some of you already know. But you need to install the add-ons. Okay. Specific for bioinformatics, there is a bioinformatics folder here, plugins, and also single cell plugins. Spectroscopy is dealing with spectroscopy data from medical devices, but you can install spectroscopy too. So once you get it, you will get database update, geodata set. Okay. Of course, you need to learn all of these widgets in YouTube to understand what is the basic use case of this bioinformatics. So focus on bioinformatics and also the single cell plugins in Orange. That can help you to get to understand basic activity in the bioinformatics. Okay. Let me continue. So International Nucleotide Sequence Database Collaboration is collaboration between three big labs and CBI, National Center of Biotech Information, European Bioinformatics Institute, and DNA Data Bank of Japan. So initially they are separated, but based on INSGC, they are combined, so they exchange data each other, making a connected and open network for archiving the data, such tools, workspace, and also external sources and analysis tools. So collecting new data for genomic is quite expensive. So for example, you take the data from saliva and blood and then go to BRIN, our research agency in Indonesia. They have many next generation sequence device there. You pay the operator there to make sure you get the data. You get the data like FASTQ file or SUM or BAM. And then you start to explore or do analysis. But for you in master degree, I think start with the available data in NCBI or EBI or the DGB. Okay, all of this connected. So as long as you can find the data, let's say COVID data for DNA for COVID or cancer or anything, you can start with public data bank. So since year of 2000, when the Sanger method is invented, people start to collect data in NCBI, EBI, then in DDGP. Yeah. So if you see here, cumulative number of human genomes increase, yeah, double every 7 months, double every 12 months for Illumina, and double every 18 months according to more laws. So data is everywhere over there. So some misconception in bioinformatics community is they want to collect Indonesian data, yeah, because they think the difference between Indonesian DNA data and other country data is quite big. But for some phenotype, maybe yes, but for other phenotype, maybe not. So as a student, you have to be very pragmatic in this case because of the cost. So I believe you have to start with the gene data, public data bank that available. So the way we're thinking in accessing data, yeah, you can because this is a very big database, yeah. If you go to INSDC website or you go directly to NCBI website or DDGB or ABI, you can search in the internet. It's a huge public data bank. How you access this? You download all? Of course, no, because you will need data center to store the data. Okay, so individual gene data itself can be 20 gigabytes or gigabytes in size. What about if you have to download million of data? So people use cloud. Like if you see in Terra Bio, you can store the data, download from the public data bank, and then store in Azure or Google bucket. And then you can do computation in Terra Bio. Okay, you can download or accessing the data as an individual genes if you know the individual genes, or some codification of the data, or proteins or molecules. Okay, but in terms of large data set, focus on the problem of interest. Let's say you want to find all variant of human globin genes, or patient moving mutation in genes, or DNA corresponding to set of 100 genes for hemoglobin function. So focus on more specific and download the data for this kind of things. But let the data stay there in the public data bank, because you need very huge storage to download for your if you download all. Okay. So types of data. So if you see the gene sequencing devices that when it works, the good thing is now that we already have same format, but however, the numbers of sequence in databanks is huge, yeah. And now we have the official name of the gene, like there is a database called each GNC, yeah, and gene names, yeah. In genenames.org, it will give official name of a gene and protein, so it can help you to search official name of the gene related to your research. Okay. Otherwise, if you don't use this kind of name, official name, it's very hard to find. Okay. And also the data is collected not only for DNA, but also the chromosome information, genomic DNA, RNA, and also the protein. And many databases being used. For example, if you want to focus on DNA, focus on GenBank, SRA, DB, GSS, DB, STGS, UniSTS, DB-SNP, yeah. For chromosome, maybe DB-FAR, genome and DB-FAR, mRNA, DBs, Unigene, GeoProfile, etc. So make sure that you know how to find and access the data. What about in Terra Bio? So later, is Fadin and Cinta here? Okay. So later, Fadin and Cinta will show you Terra Bio again, yeah? So in Terra Bio itself, you can learn about Terra Bio from the YouTube video for more video tutorial. But Terra Bio have about 107 million single cell dataset, 2.38 million study participants. It means that the patient or the participants of the study, and 65,000 Terra users around the globe. So it's a big community to start with. So please start with Terra Bio. Make sure you can find the workflow, and you can look on how they store the data and running the workflow and get the result end to end. So you can imagine the end-to-end flow if you work with other workflow engine like use Galaxy or maybe the SnakeMake and Nextflow. Okay. Another important tools that you need to also understand is genome browser. Yeah, there is many genome browser over there. In NCBI, National Library of Medicine, there is a genome data viewer, but also from Santa Cruz University, UCSC, there's also genome browsers there. So with genome browser, you can visualize the data to see some important information in the browser. Okay, so if I'm not mistaken, TerraBio use IGV, Integrative Genomic Viewer, integrated in TerraBio. But other than IGV, there is also other genome browser like Ensemble, genome browser for UCSC, and NCBI. Okay. So for your research, we in bioinformatics in UI with Professor Al Hadi, we're actually trying how we can utilize public data set like PubMed for genomic and perform some LLM analysis on combined with the functional genomic analysis. So imagine a scenario where you can crawl PubMed journal and then read the journal using OCR on PDF, and then utilize language model to learn from the papers, and then combine with your functional analysis from the data set. We call this project GeneX that currently ongoing for exploration. So I hope we can get more students working on this, yeah. But to work on this project, you need to follow deep learning course as well, because we will teach LLM there, including attention transformer, large language model in deep learning course, not in this course, yeah. So if you attend the deep learning course and you are interested in working in genomic, we are more than happy to guide you, work together in this research area, including nutrigenomic, pharmacogenomic, oncogenomic, therapeutic genomics subdomain. Okay, so that's it from me. So for the rest of 30 minutes, you can still discussing TerraBio and look on TerraBio features. If you can run one workflow, then other can run other workflows. So you can also do collaborative learning, like all of the participants of this study take one workflow and share to each other. Feel free to do that, because learning and working together is faster compared if you do yourself. Okay, that's it from me. Please continue with Fadin and Cinta.
Okay, thank you so much, Mr. Risman, for introduction to TerraBio. I will continue to explain about TerraBio.
Wait a minute, before you can also use ChatGPT if the slide is not clear, yeah? Like, for example, you can also explain about genomic data bank in NCBI, okay? So I will explain the history, you can ask all of this, yeah? Okay, and you can also ask ChatGPT like in context of NCBI gene data bank, write sample Python code to download genomic data set from NCBI. Of course, you need to understand there's some gene name, yeah, to do that. But what I want to say here is you can always use the ChatGPT for accessing data and downloading the data. Let's try. And once you download, you can use TerraBio, put in, or if you can download in the cloud, that is better, because you don't have to put in your laptop in case the file is too big, yeah? So you can install Python, Bio, okay? Okay, so Biopattern is already installed, and then you can, so you have to understand this kind of accessing numbers in the website for genomic name. Now, the file is downloaded, genomicdata.fasta, you can get it in here. Okay. So please learn and work with ChatGPT to improve your productivity. Okay, thank you. So please continue, Fadin.
Okay, thank you so much, Mr. Risman. I will continue. Let me share my screen. Okay, so last week we already learned about Terra Bio, and we already learned how to set up and get $3,000 free credit from GCP, and how we can use that to run Terra using Google Cloud Platform. And also for refreshment about what Terra Bio is. With Terra, you can access and analyze your data, share data securely, and also collaborate with anyone. Okay, so I will continue from the beginning. Okay, if you open the app.terra.bio, you will land to the main page to welcome to Terra Community Workbench. So this is the very first page that we will see for running Terra Bio. In this section, we can see few workspaces, few examples, and also browse data. So earlier, Mr. Risman already talked about the large data banks that already available online. On Terra, you can see at the hamburger section in the upper left on the right side, on the left side, and you can drop down the library, and also you can click on the data sets, and we can see in here many kind of data sets that are available, like 100 genomes data sets. And also if we scroll down, we can see data sets from Broad Institute, Broad Dataset Workspace Library, and also Human Cell Atlas, the Cancer Genome Atlas presented by the National Cancer Institute. And we can also preview the new data catalog. So if we toggle this button, we can see the new data catalog that comes to the Terra, for example, like genome aggregation database, and also target acute. This is in order to access this dataset, we have to grant it access first, but don't worry. There are many kind of external data that we can use, like 100 genomes project and human reference genomes. This is the most crucial data sets that we might use in the future, because we have to compare within our sample with the human reference genomes. Okay, also for the feature workspace, earlier we already discussed about GATK. There are lots of GATK workspace available in Terra Bio that you can learn and run the pipelines that already written in the Windows script, and we can run using the workflows in the Terra Bio. In code and workflows section contains a lot of GATK4 best practices workflows. And you can also find additional workflows that contains on Dockstore and also broad methods repository. For example, if we click on one of the GATK, it will be redirect to the Dockstore. Okay, so for now, we are going to learn about the data tables on Terra Bio. But before that, I might have announcement for you guys, because Terra is currently announced that Terra will have the scheduled maintenance on tomorrow or tonight to upgrade their dataset or databases, that maybe you guys will have some issues when running the workflows, because earlier I tried and maybe there are some parts that didn't work out well, but don't worry. The aim to run the Terra Bio, we can understand what is the Terra Bio platform features and also how to do that. So you can move to the workspace. You can click on here. And we can take a look at the public section. For this section, you can type in your keyboard in the search by keyword, but about Quickstart. And there are a lot of Quickstart tutorial that you can follow. For example, like if you want to start the Quickstart on Azure, if you have some free credits, go on and try that. But for these tutorials, we use the Google Cloud platform. And if we scroll down, there are many kinds that we already learned is about the Terra Quickstart workspaces. Terra Workflow Quickstart for last week. And for this week, we are going to use the Terra Data Tables Quickstart. You can click the 3 dots button in here and select clone. After that, you can select your building project that already been set up last week. And if the building project is already been set up, you can clone your workspace. I already cloned and I will leave it behind. Okay, so for the data tables, maybe you have to take a look at our previous works by last week. Okay, so for last week, we already run the part 1 and also the part 2. For the part 1 is we already run a pre-configured workflow on a single sample. We run how to use the workflow written in the WDLs to convert from the CRM format into the BAM format. And the second part is we already learned how to set up and run workflow to analyze two samples. And the third part is that I challenge you guys to run an additional workflow on the generated output of the set. So in each workspace, it contains all 5 sections. At the top menu is the dashboard about the workspace, the data, the analysis. If you will run your analysis using Jupyter Notebook, RStudio using Bioconductor, and also Galaxy that Pak Risman already mentioned before, and also the workflows. And also you can check on your current job history status via job history menu. Okay, so I will introduce about the data tables because today main topic about data. Okay, so in data tables, we will have this kind of UI. We can import your data, add data to this workspace, and also we can take a look of what is the data that already being imported to featured workspace that we already cloned. And also reference data, it is useful if we further want to compare our sample with the human reference genome, for example, and other data, for example, workspace data. This is already being pre-configured to this workspace. There are 3 types of data: phase A, and also the dictionary of the data. In the file, you can upload your other data. Okay, so what is that this section means? So data is, think about like Microsoft Excel or spreadsheet. It is the table like spreadsheets. And in this table, we can see that the very important for data in Terra Bio is contained for the identity or ID, and also the link to the data. For example, like this sample underscore ID, we have the my underscore sample and sat 12878, and also what is the CRM. Nah, in this CRM file, So the files are not located in the Terra platform actually. The files is located outside of the current workspaces because think about like a bucket in the cloud. We can access into that cloud to utilize into our workflows. If we select on this, we click on that. There will be a pop-up menu that contains that view this file in the Google Cloud Storage browser, and you can download for less than 1.1 0.01 sorry dollars. It is very cheap, and you can also terminal download command using this command like this. And that's pretty much it about data for refreshment. To move, we can start using the Terra data tables Quickstart that you already cloned.
Excuse
Why
me,
my-
Fadin.
Yeah.
There's question on chat. Please check it.
Oh, okay. Why my table is specimen, not sample? Okay. So for the specimen table is like this. So for the specimen table is on the Terra data table Quickstart that we are going to learn today that we will already clone, so the specimen table will pop up. Earlier, I discussed about this sample. It is from previous week to refreshment about what are we going to learn today based on last week experiment. So if you can maintain your workspaces in here, and you can check how to get the sample like before. Okay, it comes from the Terra workflows quick start that if you attend last week class, you have your workspaces in here, Terra workflow quick start. But if you haven't, you can type in quick start and clone the Terra workflow quick start in here. Because in this workspace, the Terra workflow quick start, it used the sample called the sample like this. Okay, but for now we are going to learn about the Terra data tables first. Okay, so about this workspace, we are going to learn about how to use workspace data table to organize, access, and analyze data, including sets of data in the cloud. We can learn how to run on a single entity in a table, make your own data table, run on sets of single entities, and also run on sets arrays as an inputs. Okay, moving on into the data. This is the visualization of what is the data in these workspaces. You can click on the specimen. If you see in here in the table section, there will be a list of what the data tables that already pre-configured in this workspace. And also in the parentheses, it contains 9. This means that in this specimen table, it contains of 9 rows of these specimen tables. And for the specimen set, it contains of 2 rows in the specimen set underscore set tables. Okay, like earlier I mentioned that the most important is the specimen underscore ID. It is the identification for what is your ID column, a unique ID for each distinct entity. And also this is the participant from, if we can see, the participant, it's divided into type of mouse here and human, and also our one underscore FASTQ file. This is the most important because it links into other cloud storage that contains the data sets. In this file, we use FASTQ file. So FASTQ file is the read file, raw output that comes from whole genome sequencing machines, that it is like the very first data that maybe we will working on, because it is the output that we can read from the fresh output from the sequencing machine. And if we take a look and these workflows, there are two pre-configured workflows that already built with Terra. We can use this first single input workflow. It is pretty simple, and also the second one is about sets as input workflow. To do that, we can move into the data. You can click the specimen, and you can choose a specimen to process. After expanding the specimen table, click any one of these 9 specimens that you will like to proceed. For example, I will click on the mouse and maybe the human. And you can click open selected data. And you can select using Whittle. And because we already pre-configured what our Whittle script and we put it on our workflows, we can select it maybe in the first or the second workflows. First, we are going to the first workflow is the single input workflows. Okay, so after we set everything up, we check on this pre-configured settings. To run these workflows with inputs defined by data table, it is correct because we use data tables, not on any other file paths. Select data table that comes from tables called specimens, and select data that we already choose to selected specimen, or you can also select from here. So it is pretty much up to you whether you can choose in this workflows or you can choose in data menu. And on the left side, it is the script, the Whittle script. If you want to understand about the Whittle script, you can ask ChatGPT what is it about, because bioinformatician write their scripts on Whittle or WDL. You can call it Whittle, and this is like the very simple script that the aim is to read the first read the input, yeah, which is the FASTQ file, and the output will be the first header of the first line output, which will be the FASTQ header. And if we take a look, the workflow sample to multiple output, call print sample first line, input is the FASTQ file and also the sample ID, and the output is the first line of the sample, and the task is to print the sample first line. And what is the command that they use is in here. Unzip the FASTQ file, because the FASTQ file have to be interpreted first, because it is not in the it is already in .gz format. It have to be unzip first, and the output and also the runtime. For example, we specify the CPU core will be 4, the memory and this, and also the Docker using the broad instructor JTK Docker. You can copy and select the ChatGPT for your better understanding. ChatGPT. Explain about this Whittle script below to run on Terra Bio. Okay, so ChatGPT will act like your super assistant for your better understanding about Terra Bio. If you haven't familiar about WIDL or workflow description language, that is okay. You can use ChatGPT. So the WIDL script is designed to run on Terra Bio, a platform that facilitates any analysis of biomedical data through cloud computing. The script defines a workflow named sample to multiple output that processing sequencing data to extract and output the first line of a given FASTQ file. And ChatGPT will break down of its component. For example, the workflow sample to multiple output. The input is the file and also the string sample ID, the call statement, the output, and also the task to print sample first line, which is what is the input command to decompress the input files using gunzip because the FASTQ file is using the .gz extension, and also another steps to pre-processing the FASTQ file, and the outputs will be the file that contains the sample first line. And also, like I said earlier, the runtime to specify the computational resources required for running the task, like CPU, memory, disk, and also the Docker. Okay, going back to the Terra. Okay, yeah. You can click on Run Analysis and launch. Okay, so the status will be changes. You can check within the job history menu if whether or not your workflows is running correctly or not. And also another things that I want to share with you is about this. Okay, so if you click on the link on the dashboard of the workspace, you can find the data table Quickstart Part 1 articles. And this is how the Terra works, like in the cloud, because you can think like Terra is like a computer in the cloud, and there are some buckets workspace storage act like a workspace storage that store a lot of data in the cloud. So in the data tables, we have to link what is the what is the data that we want to use that already exists in the cloud storage, not in the Terra. That is the logics of you guys understand what is the behind computation that working on behind Terra. Moving on to the data, you can also run by yourself to the second sets as input workflow, and you can select your data in the second workflows. I will expand it. There are two columns. The first column is specimen set ID, and the second column is the specimens. You can use it. Okay, and also you can run analysis, and also you can launch it. It is pretty straightforward to do that on Terra. You can practice multiple times to do that for getting familiar about the Terra. And you can maintain in the job history whether or not your workflow, your pipeline is succeed. And also Terra have another quick start that you can, I will talk about if you can type in quick start. There are 1, 2, 3, 4, 5, 1, 2, 3, I will filter using Google Cloud Platform. 1, 2, 3, 4, 5, 6, 7, 8. There are 8 quick start tutorials to run Terra Bio. For the T101, it is the pretty new, the up-to-date workflow, but it is using the GPA datasets, GPA student datasets, not the bioinformatics datasets. So you can try it on yourself because it is pretty much more easy than the Terra workflows or Terra data tables Quickstart. I'm having a problem to clone it, maybe because the Terra is facing some troubles in today. So I will move into my another account. I'm using different account. This is the data tables to quick start copy. If we take a look of this data, it contains of participant name and also sample set. You can add more rows in here. You can edit, add row, add columns in Terra. Saya pamit dulu ya. Oke, siap. Thank you so much, Mr. Risman, for attending this class today and help us understand about Terra Bio. Okay, maybe I will introduce about this workspace because I cannot run it on my account because I currently cannot clone it because having some trouble issue in Terra because of the maintenance problem. For example, in this section, we can edit the data like in this in the participant, I think it is more. We can add row participant ID. Maybe if we do some research and we want to add a new row to this column, we can maybe type in what is the participant ID about, and it will add it to the workspace, to the data section in the workspace. And also you can select rows in the data tables and can export the selected data into a workspace, copy or download as TSV. And also you can there are advanced search like search logic and all. And here is the workspace that contains human reference data that I talked earlier about the human genome. So maybe next week we are going to learn about how we can use human reference data to do our analysis. So that's pretty much it. It is pretty straightforward. The aims of this current session is to getting familiar about data tables, also the dashboard analysis, and also the job history in the Terra Bio. For the analysis, you can click on, maybe I will check in the my current if there is any. Okay. If there are, okay. So I will introduce about the analysis. Hopefully it will cover up everything that is available on Terra. To analysis like Jupyter Notebook, if you guys familiar about it, you can use Jupyter Notebooks on Terra. I will click on it. It is the preview read only. Hopefully it can, okay, edit it. Right. Unfortunately, it cannot be seen. Maybe I will check in another. Maybe I will introduce about notebook. Okay. Okay. So you can code your Python code in this workflow section analysis menu in the Terra Bio workspaces. Like you can pretty much like type in your Python code with using data in the Terra Bio workspaces. Unfortunately, I am having problem to clone it, so maybe I cannot show you guys how to run it, but I will take a look about what is going on in the analysis. So pretty much on analysis, you can start using Jupyter Notebook, using Galaxy, and also using ours for bioconductor. And also in this Jupyter Notebooks one-on-one, for example, to do this, to run this notebook, you can select or set up your cloud environment VM in this section first. And also, like you guys did on maybe if you familiar about Google Colab, you can run and analyzes your data using Jupyter Notebook in here. Yeah, it's Terra issue. I got the same error too. Yeah, unfortunately, maybe for today, there will be much, you guys will be facing errors when you run the workspaces, I think, because I'm having a trouble in today to run the process smoothly, but maybe in the next week I will talk about this exciting workspaces that we will talk. So it is getting pretty much next level because in today we already talked about the quick start section, about the data tables, about the notebook, and also about the workflows. For the next section, the next week section, we will talk about the first GATK workspaces, which is the sequence format conversion. So in order to read the bioinformatics or the genomics sample that already proceed by genomes machine by like Illumina, if you guys familiar about it. This is the workspace that we have to take a look first. How to use FastQ and also BAM, CRAM to BAM, VCF. That is pretty much it because in the bioinformatics, I will talk about this. Okay. Okay, so in bioinformatics is divided into three sections. First is the primary analysis, second one is secondary analysis, and the third one, of course, is the tertiary analysis. So for the first primary is pretty much for the library preparation, sequencer, QC check. It is pretty much about on the software, on the hardware machines, the like Illumina. And after the instrument reads about our sample, the FASTQ format will be generated like here. This is the area that bioinformatician will be covered up most about in the secondary for the alignment, QC check, and also the variant calling. Okay, here's the pipeline for the primary analysis for sequence generation, signal analysis, base calling, base quality scoring, read generation FASTQ file. It is pretty much about the reading. And after that, we can continue to secondary analysis, sequence processing using human reference genome that already contains from many years before. And we do that into alignment to genome BAM file from FASTQ file, and also base quality score, recalibration, and for at the end is variant calling file that we want to use for upcoming tertiary analysis for results interpretation. We will cover up about the next section about this. Hopefully you guys getting more familiar about bioinformatics and also for Terra. Like Terra Bio, Nextflow, SnakeMake, Galaxy project, and also another any kind of tools. It is pretty much like a pretty bioinformatician platform that we use, but we have to understand first of what is going on instead of just do that, just click on the Terra Bio. So it is much more than that. We have to understand first about what is going on in the bioinformatician side of the pipeline inside the Terra Bio. That's pretty much it. We will continue on the next section about these workspaces into sequence format conversion. Hopefully we can run smoothly next week and Terra Bio, and also in Terra Bio, and also maybe we can moving on into Galaxy, Nextflow, SnakeMake, according to the materials that Mr. Risman will cover up into the next week. If you guys have any question, feel free to ask in the column, the chat box, and also you can check into your mic to talk about what is your issue. Feel free to ask. Thank you. Okay. For the record, maybe Mbak Sinta will help to distribute it, the previous record that already being edited to you, Mbak Sinta.
Sure, I will share it into YouTube Machine Learning Indonesia. So please stand by there, everybody.
Okay. So feel free to reach out to me via WhatsApp on personal. I'm very glad to be able to help you guys. And thank you for attending this class. We continue again next week and to the next topic. Thank you so much.
Thank you,
Thank
Mbak
you.
Fadin. Thank you, everybody.
Thank you.